Today we are going to talk about polymers, which are chemicals made by living organisms. Polymers are large molecules, also called macromolecules. The term macro means large. Biological macromolecules can be classified into four major types. Proteins, also known as polypeptides. Polypeptides consist of many amino acids linked together. Carbohydrates or polysaccharides. Saccharides means sugar and poly means many, so carbohydrates are made up of many simple sugars. Lipids, which include oil, fat, waxes, and steroids. Most lipids are made from glycerol and fatty acids, though, as you will learn today, steroids are a unique subgroup of lipids. Nucleic acids, DNA, RNA, and ATP. We will leave nucleic acids when we will cover genetics in a later unit. Today we will focus on three macromolecule groups: proteins, carbohydrates, and lipids. Some organic molecules are small and called monomers. Mono means one and mer means part. So monomers are single unit molecules. For example, methane is a simple organic compound consisting of one carbon atom and four hydrogen atoms. Monomers can link together to form polymers, which are lone molecules made up of many monomers. In this image, we see the structure of fat molecule called triglyceride. It has glycerol head and three fatty acid tails. We'll discuss fatty acids later in this lecture. Okay, once again, all macromolecules are polymers, are large molecules formed by joining smaller subunits together called monomers. One way monomers link together is through a condensation reaction. In this process, monomers lose hydrogen and oxygen atoms which form water, creating bonding sites that allow monomers to join. This reaction is also known as dehydration synthesis because water is removed during the process. Here is a visual representation of a polymer. Notice one end has a hydroxyl group and the other end a hydrogen. It is important not to confuse the hydroxyl group with the hydroxide ion, which we have discussed earlier as part of dissociation reactions in water. The hydroxyl group is the functional group attached via covalent bonds. Now let's say we want to extend this short polymer. If it is a protein, the monomer we will add is an amino acid. If it is a carbohydrate, it will be a simple sugar. Enzymes are required for this reaction. Enzymes are biological catalysts that speed up biochemical reactions. They will help us to link the monomer to the polymer by removing a water molecule. A dehydration reaction. The opposite process is hydrolysis, a catabolic reaction in which water is used to break down polymers into monomers. You see, here we have water molecule. It gets in between two monomers. As a result, we end up with a shorter polymer and a monomer. This is hydrolysis or hydrolytic reaction. As you already know, hydro means water, but think of hydrolysis as a word for breaking. It is a catabolic reaction, unlike dehydration reaction or condensation reaction, which is anabolic. For example, if we have a disaccharide, adding water breaks it into two monosaccharides or two simple sugars. It hydrolyzes molecule of disaccharide into two smaller molecules. This is what we call hydrolysis. Here is another example, hydrolysis of lactose. A carbohydrate disaccharide, lactose, was hydrolyzed and we ended up with its monomers. Simple sugar galactose and simple sugar glucose are smaller molecules. Note that this quick reaction will not be possible without an enzyme called lactase. People who are lactose intolerant typically produce insufficient amount of the enzyme lactase, which is needed to break down lactose, the sugar found in milk and dairy products. This deficiency leads to difficulties in digesting lactose, causing problems like bloating, gas, and diarrhea. In this slide, you see symbolic representations of lactose on its monomers. Here are more details if you are interested. Below are structural formulas of these chemicals, all of which are composed of carbon, hydrogen, and oxygen atoms. Here you see the opposite process, an anabolic reaction that we call dehydration synthesis or condensation reaction if you like, in which monomers like glucose and fructose combine, releasing water molecule and forming a larger molecule, sucrose. For simplicity, I have used symbols such as hexagons with hydroxyl groups on the side instead of structural formulas. Now let's take a look at carbohydrates. Carbohydrates, as you perhaps noticed on the previous slides, contain carbon, hydrogen, and oxygen. The energy stored in the covalent bonds between carbon and hydrogen is released when organisms break these bonds. In fact, this is how organisms, including humans, get their energy from the food they eat. The energy comes from the breakage of covalent bonds. Carbohydrates are divided into three types. Monosaccharides, simple sugars like glucose. Disaccharides, two monosaccharides linked together like sucrose, table sugar, and lactose. And polysaccharides, long chains of monosaccharides such as starch, cellulose, and glycogen. Monosaccharides and disaccharides are crystalline, sweet, water-soluble, and have names ending in "ose", glucose, fructose, arabinose, sucrose, and so on. Polysaccharides like starch are not sweet and must be broken down through hydrolysis before their sugar can be absorbed. Polysaccharides are composed of long chains of monosaccharide units. I hope you understand that the wavy lines indicated by the red arrows in the structural formula of glucose represent the continuation of the same repeating monomers. Polysaccharides like cellulose are also known as dietary fiber in our diet. While humans cannot digest cellulose because we lack necessary enzymes, it plays a crucial role in digestive health. It adds bulk to stool and aids in regulating bowel movements, keeping the digestive system functioning smoothly. Beta-glucose is a monomer unit in cellulose. As a result of the bond angle in the beta-acetyl linkage, cellulose is mostly a linear chain. Lack of dietary fiber can lead to several health issues, including constipation, where stool becomes hard and difficult to pass. Frequent constipation can lead to increased risk of developing hemorrhoids, which are swollen veins in the anus. A more serious condition called diverticulosis, where small pouches form in the colon wall. These pouches can become inflamed, diverticulitis, leading to pain and potentially requiring surgery. In contrast, a diet rich in fiber helps prevent these issues and supports overall digestive health. Another carbohydrate protein that we cannot digest is chitin, a derivative of glucose and is a characteristic component of cell walls of fungi and the exoskeletons of crustaceans. Chitin is a polysaccharide containing nitrogen. Chitin belongs to the biopolymer group, and as you can see on this slide, its fibrous structure is similar to cellulose. While humans cannot digest chitin, eating foods that contain chitin, such as mushrooms, can still provide some benefits for digestive health. Chitin acts similarly to dietary fiber, which means it can help promote bowel movements by adding bulk to stool. That said, chitin's primary role in diets is limited. It can offer mild digestive benefits in terms of promoting intestinal movement, but it does not significantly impact digestive health in a way that other types of fiber-rich foods like fruits, vegetables, and whole grains do. More research is needed to fully understand chitin's effects on human digestion. In summary, two polysaccharides that we as humans cannot digest but still important in our diet are cellulose and chitin. Now let's take a look at the carbohydrates polymers that we digest. Starch is a polysaccharide made up of glucose molecules. It is found in foods like potato, rice, wheat, and corn. Starch is made up of long chains of glucose units. It is broken down by enzymes in the digestive system into glucose, which is then absorbed into the bloodstream and used as a fuel by the body. Another polysaccharide that is digestible by us humans is glycogen. It is found mainly in the liver and muscles and can be broken down into glucose when energy is needed. On this, we finished with carbohydrates. The next group of polymers is proteins. Proteins are essential macromolecules found in every cell. Proteins constitute over 50% of cell dry weight. They made up of amino acid subunits. They are known to be most versatile molecules. They are composed of amino acids which link together by peptide bonds. Peptide bonds are a specific type of covalent bonds. This term is used to specify that we are referring to the covalent bonds that link amino acids together in proteins. The sequence and number of amino acids determines the shape and function of protein. There are 20 different amino acids that are commonly found in protein, and their sequence dictates how protein folds and functions. Proteins serve many vital roles, including structural support, giving shape to cell and tissues, catalyzing reactions, enzymes speed up biochemical reactions, gene regulation, protein help control gene expression, nutrient acquisition, protein aid in obtaining nutrients necessary for growth and maintenance. Amino acids have a central carbon, an amino group, a carboxyl group, and the unique side chain that differentiate one amino acid from another. Amino acid can be classified based on the nature of their side chain. Let's take a look at non-polar aliphatic R groups. This includes amino acids like glycine, alanine, leucine, and others. These side chains do not carry a charge and are hydrophobic, meaning they repel water and do not dissolve in it. These chains form linear structures or aliphatic structures, as opposite to cyclic structures. Polar and charged R groups examples include serine and glutamine. These side chains have slight charges due to atoms like oxygen, making them hydrophilic, water attracting, though they are not fully charged. The presence of atoms like oxygen pulls electrons, creating polarity. Look at positively charged R groups. These include lysine, arginine, and histidine, which have a positive charge due to the presence of extra hydrogen atoms. These side chains are ionic, meaning they interact with other charged molecules. Look at negatively charged R groups. Aspartate and glutamate have negatively charged side chains, which are also ionic. And here non-polar aromatic R groups. This group contain amino acids like phenylalanine, tyrosine, and tryptophan, which have aromatic rings in their side chain, making them distinct from other groups. Proteins fall into specific shape based on their amino acid sequence, and this structure is organized into four levels: primary, secondary, tertiary, and quaternary. Primary structure. This is the linear sequence of amino acid in the protein, which ultimately dictates how the protein will fold. Of course, it has amino terminal and carboxyl terminal end. And here you see different amino acids which may or may not repeat. In secondary structure, the amino acid change falls into repeating patterns such as alpha helices, spiral shapes, and beta-plated sheets, folded sheets. These structures are stabilized by hydrogen bonds between different parts of the chain, somewhat like this. Tertiary structure is the overall 3D shape of a protein formed by interactions between the R groups. As you see, bonds involved include hydrogen bonds, ionic bonds, disulfide bridges, covalent bonds between sulfur atoms, and hydrophobic interactions. Okay, now we have to understand the quaternary structure. You see, some proteins are made of multiple folded subunits, tertiary structures that come together to form functional protein complex like hemoglobin, which has 4 subunits. The quaternary structure determines how these subunits interact and function together. The specific shape of a protein is essential to its function. For example, enzymes. Enzymes are proteins. They are catalysts, meaning that they make a reaction go faster, but enzymes themselves are not altered by overall reaction. They must bind to a specific molecules called substrates. The enzyme's active site fits the substrate like a lock and a key, ensuring that the correct chemical reaction occurs. Proteins require specific shape to function properly. If the protein loses its shape due to its external factors like heat or pH changes, it can become denaturated and unable to perform its function. Denaturation is the process in which protein lose its quaternary, tertiary, and secondary structures which are essential for their native function. This can occur due to external stress or compounds such as strong acids or bases concentrated in organic salts or organic solvents. Denaturation is the process in which a protein unfolds and loses its natural shape due to stress like heat, acids, or bases. This can be irreversible, as in case of cooking an egg, where the heat denaturates the protein, causing the egg to solidify. Lipids are a large group of compounds that are insoluble in water. They include fats, phospholipids, and steroids, and they contain carbon, hydrogen, and oxygen, but with less oxygen than carbohydrates. Lipids serve many important roles in the body, such as energy storage. Lipids provide a dense source of energy. Hormone regulation, steroid hormones derived from lipids, they regulate various body processes and other functions are cushioning and protection. Lipids protect vital organs by cushioning them. And the last but not the least lipid function is transport of fat-soluble nutrients. Vitamins like A, D, E, and K are transported by lipids. The calorie density scale is a tool used to measure the number of calories in a given weight of food, typically expressed as calories per gram or calories per pound. It helps to categorize food based on how energy dense they are, which can guide individuals toward healthier eating patterns. Understanding calorie density can be useful to weight management, as it allows you to eat larger portions of lower calorie foods, which are more filling and nutritious without consuming excessive calories. The calorie density scale measures how many calories are in the given weight of food. As you can see, oils are highly calorie dense, containing around 4,000 calories per pound, while nuts also have high calorie density at about 2,800 calories per pound. In contrast, vegetables around 100 calories per pound, fruits around 300 calories per pound, and potatoes around 400 calories per pound are much lower in calorie density. Pasta, rice, beans, and peas have moderate calorie density, 500 to 700 calories per pound, making them filling yet more energy dense than vegetables, but much lower in calories than oils or nuts. In the lipids dehydration reaction, fatty acids and glycerol molecules combine to form triglyceride, a type of fat, through a dehydration synthesis. During this reaction, each fatty acid molecule forms a bond with the glycerol backbone by removing a molecule of water. The hydroxyl group from the glycerol and hydrogen atom from the fatty acids are removed, resulting in the release of 3 water molecules, one for each fatty acid. This process creates the ester bonds that link the fatty acids to the glycerol, forming a triglyceride. Triglycerides in our diet are the main form of fat found in foods like oil, butter, and meat. Once consumed, they are broken down during digestion and either used for the energy or stored as a body fat. Saturated fats found in butter, coconut oil, etc., containing no double bonds in their carbon chains, making them straight molecules. They are solid at room temperature and are more resistant to oxidation, which is why they are often used for cooking. However, consuming too many saturated fats can increase bad cholesterol levels, which is linked to heart disease. The majority of cholesterol in the body, about 80 to 90%, is produced by the liver, while only 10 to 20% comes from dietary sources. Saturated fats can influence the liver's cholesterol production, particularly in people who are more sensitive to dietary fats. However, cholesterol levels are primarily regulated by the body's own production, which is influenced by the factors such as genetics, hormones, and overall health. So the emphasis on dietary cholesterol is less impactful than once thought for most people, as their liver compensates by adjusting its own cholesterol production. So why do nutritionists recommend limiting the consumption of foods high in saturated fats? Nutritionists recommend limiting the consumption of foods high in saturated fats because these fats can influence the liver to produce more LDL, bad cholesterol, which may increase the risk of heart disease. Although only about 10 to 20% of cholesterol comes from diet, saturated fats can still affect the body's cholesterol balance, especially in those sensitive to them. Cholesterol has gained a negative reputation because high levels of LDL, low-density lipoprotein cholesterol, can lead to buildup of fatty deposits, plaque in the arteries, increasing the risk of heart disease and stroke. However, HDL, high-density lipoprotein cholesterol, is beneficial because it helps transport excess cholesterol from the bloodstream back to the liver, where it is broken down and eliminated. While dietary cholesterol from foods like egg yolk, meat, and dairy contributes to overall cholesterol level, the majority of cholesterol in the body is produced by the liver. Genetic factors, lifestyle choices, and dietary habits all influence cholesterol level, but saturated fats and trans fats in the diet tend to have a greater impact on increasing LDL cholesterol than dietary cholesterol itself. Which foods are high in cholesterol? Animal-based foods such as egg yolk, red meat, and cream are high in cholesterol. Additionally, shellfish like oysters, mussels, crab, lobsters, and clams contain significant amount of cholesterol, with squid having the highest cholesterol content per unit weight among seafood. Does most of the cholesterol in our blood come from our diet? No, most of the cholesterol in our blood is not directly from our diet. Among 80 to 90% of circulating cholesterol is produced by the liver, with only a small portion coming from the food we consume. Now, what does fully saturated with hydrogen means? Fully saturated with hydrogen refers to a fatty acid in which all the carbon atoms in the chain are bounded to the maximum number of hydrogen atoms. This means there are no double bonds between the carbon atoms, making the fatty acid straight and saturated with hydrogen. Saturated fats are typically solid at room temperature, like butter or lard. Now, what does highly resistant to oxidation means? Highly resistant to oxidation means that a substance such as saturated fat does not easily react with oxygen. This reduces the likelihood of substance breaking down or forming harmful compounds like free radicals. In the context of fats, being resistant to oxidation helps prevent spoilage and formation of potentially damaging oxidative byproducts during cooking or storage. Oxidation occurs when molecules lose electrons, often creating unstable free radicals. Free radicals are unstable molecules or atoms that have an unpaired electron, making them highly reactive. In the body, they can steal electrons from other molecules, leading to cellular damage. Free radicals are produced naturally during processes like metabolism, but excessive free radicals can contribute to disease such as cancer, heart disease, and aging. Antioxidants help neutralize free radicals by donating electrons, thus reducing their harmful effects. Here is an example of hydroxyl radical. Hydroxyl radicals are extremely reactive and can attack a wide variety of organic molecules. Their strong oxidizing properties are due to their high oxidation potential, making them one of the most aggressive oxidants in nature. So, saturated fats are highly resistant to oxidation and therefore do not form dangerous free radicals like many oils do. Fatty acids that contain one or more double bonds in their carbon chain are referred to as unsaturated fats. Here, notice the presence of one double bond. In place of hydrogen, the molecule forms a double bond, creating a kink in the fatty acid chain. Unlike saturated fats, which have straight chains, the kink makes the molecule bend. When there is only one double bond and therefore one kink in the lipid molecule, it is called monounsaturated fatty acid. Mono means one, referring to the one double bond or kink in the fatty acid chain. This kink prevents the molecules from packing closely together, which is why monounsaturated oils are liquid at room temperature. In contrast, saturated fats have straight molecules that can pack tightly, making fats like butter and lard solid at room temperature. Monounsaturated fats found in olive oil and canola oil are highly resistant to oxidation, meaning they produce fewer free radicals compared to other oils. This makes them a healthier choice for cooking and consumption. Also, monounsaturated fats are generally considered heart healthy because they can help lower bad cholesterol levels while maintaining or even increasing good cholesterol levels. This improves overall cholesterol balance, reducing the risk of plaque buildup in the arteries and promoting better heart health. Polyunsaturated fats found in sources like fish oil, safflower oil, sunflower oil, corn oil, and cottonseed oil have the following characteristics. They lack hydrogen atoms at multiple points in their structure due to the presence of two or more double bonds. Here at the bottom of the slide is polyunsaturated fat. Notice that it has multiple double bonds in its fatty acid chain, which is why it is called polyunsaturated. These double bonds create kinks in the structure, preventing the fat molecules from packing tightly together. The good thing about polyunsaturated fats is that they can help lower bad cholesterol levels in the body, lowering the risk of heart disease and stroke. Omega-3 fatty acids found in polyunsaturated oils help reduce inflammation, which can lower the risk of chronic diseases like arthritis. Omega-3s are essential for brain health, improving cognitive function and reducing the risk of neurodegenerative diseases. The downside of polyunsaturated oils is their susceptibility to oxidation, especially when they are exposed to heat, light, or air. This can lead to the formation of harmful compounds like free radicals, which may damage cells and contribute to inflammation and chronic diseases, including cancer and heart disease. For this reason, polyunsaturated oils are not ideal for high-heat cooking, such as frying, because they can break down and lose their health benefits. Additionally, an excess of omega-6 fatty acids commonly found in these oils can promote inflammation if not balanced with enough omega-3s in the diet. Phospholipids are major components of cell membranes, playing a crucial role in maintaining the structure and function of cells. These molecules have a dual nature. They have hydrophilic head. This part of the molecule is attracted to water polar and consists of phosphate group. Hydrophobic tails. These are two fatty acid chains that repel water, non-polar. Phospholipids are amphipathic molecules, meaning they contain both hydrophilic and hydrophobic parts. When placed in water, phospholipids naturally arrange themselves into a bilayer, with the hydrophilic heads facing outward towards the water and hydrophobic tails facing inward, away from the water. This arrangement forms the basic structure of all cell membranes, creating a barrier that separates the interior of the cell from the external environment. The amphipathic nature of phospholipid is crucial for the formation of the cell membrane and allows for selective permeability, meaning that some substances can pass through the membrane while others cannot. The phospholipid bilayer also provides fluidity to the membrane, allowing proteins and other molecules to move within the membrane, which is essential for cell signaling, transport, and communication. The structure of phospholipid allowed the cell to maintain its integrity in an aqueous environment, while also enabling the transport of nutrients, waste, and signals in and out of the cell. This is why phospholipids are considered one of the most essential molecules for life. Membrane fluidity is influenced by the length of the fatty acid chains. Shorter chains increase fluidity, while longer chains decrease it. Also, membrane fluidity is influenced by the presence of unsaturated fatty acids. Double bonds in the tails create kinks, which prevent the fatty acids from packing tightly, thereby increasing membrane fluidity. Besides that, the amount of cholesterol also plays an important role in keeping cell membrane fluid. Cholesterol modulate membrane fluidity by preventing the fatty acid tails from packing too closely in cold temperatures and maintaining structure in warm temperatures. Cholesterol belong to a special group of lipids called sterols. Sterols are a subgroup of steroids. Steroids are a type of lipid characterized by co-structure of 4 fused carbon rings. Unlike other lipids, steroids do not contain glycerol or fatty acids. Instead, their biological activity is determined by the functional groups attached to the rings. One of the most well-known steroids is cholesterol, which plays multiple roles in body. Structural component of the cell membranes, cholesterol is embedded within the phospholipid bilayer, where it helps regulate membrane fluidity and stability. As I said earlier, it keeps membranes firm in high temperatures and prevents them from becoming too rigid in cold temperatures. Cholesterol is amphipathic like phospholipids, meaning it has both hydrophilic, the hydroxyl group, and hydrophobic, the steroid ring structure regions. This property allows cholesterol to integrate into cell membranes, where it contributes to the proper function of the membrane and prevents it from becoming either too fluid or too stiff. Cholesterol is precursor to steroid hormones. Cholesterol serves as a building block for the synthesis of important hormones, including testosterone, estrogen, cortisol, and vitamin D. Testosterone is the primary male sex hormone responsible for promoting muscle growth, bone mass, and development of male reproductive tissues. Estrogen is the primary female sex hormone involved in the regulation of menstrual cycle, reproductive development, and various other functions in both males and females. Testosterone is primarily produced in the testes in males and in small amounts in the adrenal glands in both males and females. Estrogen is primarily produced in the ovaries in females with additional production in the adrenal glands and fat tissues. In males, small amounts of estrogen are produced in the testes and adrenal glands. During puberty, increased estrogen level in females lead to breast development, the growth of pubic hair, and the widening of hips. In males, elevated testosterone level results in the growth of body hair, a deepening of the voice, and muscle development. The symbol for men is typically represented by a circle with an arrow pointing outward from the top right, which is associated with the male gender. This symbol is also linked to the planet Mars and the Roman god of war. The symbol for woman is typically represented by a circle with a small cross extending downwards, which is associated with the female gender. This symbol is also linked to the planet Venus and the Roman goddess of love and beauty. Now let's consider the following question. Why do some athletes take synthetic variations of testosterone? Take a moment to pause the video and reflect on your answer. As you just learned, testosterone plays a key role in stimulating muscle growth and enhancing physical performance. Some athletes take synthetic variations of testosterone known as anabolic steroids to accelerate muscle development, increase strength, and improve endurance beyond their natural capacity. However, this practice can lead to serious health risks and is banned in mass sports due to its unfair performance-enhancing effects. But what are potential side effects of testosterone supplements? One of them is increased aggression, often referred to as a roid rage. Another is elevated cholesterol levels. Increased risk of certain cancers, particularly with long-term use. Nearly all athletic organizations have banned the use of testosterone supplements. In conclusion, polymers, specifically biological macromolecules, are vital for life, playing an essential role in structure, function, and energy processes within the living organisms. We explore the main classes of these macromolecules: proteins, carbohydrates, and lipids, each of which serves distinct yet interconnected functions. Proteins built from amino acids contribute to cellular structure, catalyze biochemical reactions as enzymes, and regulate genes expression. Carbohydrates provide a primary energy source and serve as a structural elements with complex forms like polysaccharides, adding dietary fiber crucial for digestive health. Lipids encompassing fats, oils, and steroids are important for long-term energy storage, hormone regulation, and cellular membrane integrity. The formation of these macromolecules occurs through dehydration synthesis, where monomers link by releasing water, while their breakdown involves hydrolysis, utilizing water to split them back into monomers. This anabolic and catabolic balance highlights the intricate biochemical pathways essential to maintaining life processes. Each macromolecule class, from the energy-dense carbohydrates to versatile proteins and hydrophobic lipids, reflects diversity and specialization within the biological systems, demonstrating how these polymers are not only building blocks of life, but also critical to maintaining cellular and systemic functions in organisms. Understanding polymers and their roles provides insights into metabolic processes, dietary needs, and even genetic expressions, offering a foundation for exploring more complex biological topics such as genetics, cellular respiration, and metabolism.